right, good morning, good afternoon, whatever it may be. This is the Biology Quarter 3 exam review. So I'll get right into it. The first thing we talk about is viruses. So some things to remember about viruses. Remember that viruses are really small. Okay, how small? They're like 1 to 100 or 1,000 nanometers. So remember, a nanometer is 1/1000th of a meter. So nanometer is 1/1000th of a meter. Okay, they're basically a shapeless thing. Remember, they can have any shape. But they did have 4 specific parts. If you remember, those parts is a glycoprotein. And remember, the glycoprotein is the thing that it can use to change its shape and make its identity, and it can use it to get in and out of cells and trick cells into getting in there. So this is that ID tag that we talked about a long time ago with the cell membrane. Okay, it has DNA or RNA. So whatever genetic material that it has, some have DNA, some have RNA. That's found in the middle, but there is no nucleus. So it's just this, particle. So a virus is a particle. It's not a cell. And out here we have a lipid coat or a lipid envelope. And the way I tell this apart is I just look at this, and this picture doesn't do it justice, but if you look at yours, you'll see like how it's got the phospholipid bilayer, just like in the cell membrane. That tells you that it's a lipid, right? It's a phospholipid. And then lastly, we have a protein coat that obviously does some sort of protective layer here and here around the virus. Okay, so we know that they're any shape, but they have 4 specific parts to them. So, if you had to define a virus, it's a small particle that is not living, that is made of, lipids and proteins that surround DNA, basically is what a definition would be. Okay, we talked about some specific viruses in our class. We talked about HIV, talked about Ebola, we talked about H1N1, and we talked about bacteriophage. So, just kind of review some of the stuff about them. Remember, HIV was a circular virus, kind of like this one right here, that caused AIDS. And so basically, transmission of HIV is done through, bodily fluids, blood, things like that. Humans and some primates are hosts, and it causes a deficiency in your immune system. Okay, Ebola, remember, is this little snaky-shaped one, and it leads to a disease called hemorrhagic fever. And so remember, that causes internal bleeding and causes all of your internal organs to liquefy. Hemorrhaging, that's what hemorrhaging is, bleeding. Bats have been implicated as one of the hosts, and obviously us. And it's transmitted through liquids in the air, sneezing, coughing, things like that. Okay? Some people get it through the soil and stuff like that. Okay? H1N1 is another circular one like this one right here. So that also looks like this. These two right here look like this. Okay, H1N1 is also called the swine flu. And so remember, swine flu is basically influenza that has evolved from originally a bird flu, which then went to pigs and humans. So it's a bird, pig, human flu. Those are the 3 hosts. And it causes, flu-like symptoms, headache and vomiting and fever and stuff like that. That is passed on from person to person, coughing, sneezing, bodily fluids, those types of issues. Okay, and the last one was bacteriophage. Remember, bacteriophage is that weird rocket-shaped thing that looked like a spaceship. Remember, this one didn't really cause disease. It only affected bacteria. So there's no real disease name for this, but it infects bacteria and kills bacterial cells. Okay, so those are the 4 viruses. A little bit about how viruses are transmitted. So yeah. Okay, this is how viruses reproduce. Okay, so viruses reproduce 2 ways. They are called the lytic and the lysogenic cycle. So this top one is the lytic, and the bottom one is the lysogenic. And if you remember, we learned these separately and then stuck them together. So I'll kind of go through them separately. So the lytic cycle is where basically viruses are going to make more viruses. Okay. Now they don't actually make them. They make other cells. Remember, they trick other cells into making them. In this case, it's probably an E. coli here. But basically, it's viruses making more viruses. Okay. So if you go through these, you can follow along in your sheet or look at your sheet. Here's where it's going to inject its DNA. So it infects it, injects it. Okay, so here's its DNA. That DNA is going to go over here and assimilate with the host DNA right here. So you can see a little chunk right here that is actually virus DNA. So now the host has virus DNA with its own DNA. Okay, so it's going to go over here. It's going to start assembling or making parts. Okay, and it's going to assemble those parts. Okay, and then over here is where it's going to lyse. So this is where the word comes from, lyse. So it's going to lyse, it's going to burst this cell, it's going to break, these are going to build up inside, and it's eventually going to pop, and these viruses are going to be released, and they're going to look for a new host, and they're going to repeat the process. And so if you're in the lytic cycle, if you're the host, you're probably showing symptoms of being infected because you're losing cells. You're getting your cells destroyed. It's killing cells. Okay. If you're at the lysogenic cycle, that's different. You're not going to show symptoms. Okay, because this one quite simply is just viruses make more DNA. Okay, and again, they're not making it. The host is making it for them, but they're making more DNA or RNA. So here, the injection happened. Here, the assimilation happens where the DNA goes together. Here, we're doing what's called replication. We're copying the DNA. Remember, we talked about that with binary fission. So here we have 2 copies of the host DNA. Again, it's got pieces of virus DNA attached to it. Here, this cell is dividing, okay? But when it divides, it's going to make new cells with DNA that has viral DNA inside it. So the host always has that viral DNA as part of its DNA, and if your resources get good enough to support this, then they'll switch over and go lysogenic, and if not, you'll continue to just do lysogenic, and you'll be fine. Okay, so this is about resources, really. If you have the low resources, you'll do this. If you have good resources, it'll switch over and it'll do lytic cycle. Okay? Remember that viruses cannot reproduce without a host. They need a host with the most. Okay, so that's everything I can think of about the viruses. There's 15 questions about viruses, all multiple choice, so you'll be able to read that picture and label that other picture for sure. Those are both on there. Okay, this is about respiration. So respiration, remember that respiration is eating and breathing to make energy. Okay, eating and breathing to make energy. So respiration happens in your mitochondria. Okay, so remember we have to get the oxygen into our lungs, into our alveoli, into our blood. We got to get our food into our stomach, into our intestines, everything's got to get to our cells. Okay, so we have to get all the stuff into the cells. Okay, once we do that, we can do respiration. So remember that all of our food, proteins and sugars and fats, and all our foods are converted into a sugar called glucose to be used, or they're converted into fat for storage. Okay, so once we get all our food turned into glucose, we can do respiration. So remember that we eat glucose, which is C6H12O6. We breathe oxygen, so that's 6O2. And we make energy, and our cell energy is ATP, so we make 36 of those. We make carbon dioxide, which is CO2, we make 6 of those. And we make 6 H2Os, which is water. Okay, so here's the formula for respiration right there. Here's your reactants. Here's your products. And so the mitochondria is significant. Remember that the shape of the mitochondria, we've always just drawn it like this with the little foldy things. The little foldy things are membranes. And those membranes are important because they increase the surface area of the mitochondria itself. And that is where stage 3 happens. That's where the actual ATP are made, the energy. So they're going to put hydrogen ions through those membranes to make ATP. So by having more membranes, you're going to make more ATP. All right. Okay, first stage of respiration is called glycolysis. And glycolysis quite simply takes glucose, a sugar, and converts it into a smaller sugar called pyruvate. So really, in a sense, we're splitting glucose in half. Okay, it's essentially what you're doing. Okay, now it takes a little bit of energy to break bonds to split this thing in half, but when we do that, we are able to then double our energy that we put in and make out of it. So reactants are glucose. Reactants are glucose and NAD and ADP, which are not necessarily of concern, but nonetheless they are. Okay, and actually we do use a little bit, we do use a little bit of ATP initially also. Okay, so just, I might ask you about a section of this, so just read the graph. So here's a reactant, here's a reactant, here's a reactant, here's a main reactant. Okay, your products, what you get out of it, yeah, you get this ADP, but it's then used again later. Okay, but our real products, we get NADH, and we get ATP, and we get pyruvate. So that's the main one, is that pyruvate. You might call it pyruvic acid, depending on what picture you have. So again, just read it. Remember that we also put in 2 ATP, but we got out 4 ATPs, so we get out 2 ATP also. So we get a little bit of energy from doing this. Just by splitting our food in half, we get a little bit of energy. So that's glycolysis. Remember that glycolysis happens in the liquid of the mitochondria. So out in that liquid, remember there's enzymes out there. Okay? And so it's happening just outside in the mitochondria. Okay, this is called the Krebs cycle. This is stage 2. So the Krebs cycle takes what's left of glycolysis, which was pyruvate, which was 3 carbons, and it's going to stick it in this wheel and use enzymes that are found in the mitochondria itself, okay? And they're going to slowly break this thing down. So each time they go through, they're going to take one of these C's and knock it off, and they're going to form CO2. And one of these C's and knock it off, and they're going to form CO2. And so for each pyruvate, this thing's going to spin 2 times. So you're going to spin, more than once for a whole sugar molecule. You're going to spin 6 times for a whole sugar, okay? So the big thing is, what do you get out of it? This is basically converting pyruvate, I spelled that wrong, into CO2 and NADH and FADH2. Okay, so here's my main reactant right here. I have other reactants that are in here that are, again, are important, but we just kind of aren't going to talk about them too much. Here and here and here, NAD and FAD, are used to make NADH and FADH. Okay. Our main products, though, are CO2, NADH, a little bit of ATP, again CO2, NADH, and FADH2. So remember, anything after an arrow is a product. So this is going to spin essentially 3 times for this, so 6 times for glucose. Okay, and so we're going to release 6 CO2s. We're going to make 2 ATPs, and we're going to make lots of NADH and FADH, which are important. Okay, so again, this happens in the mitochondria, out here in the liquid part. Okay, last stage is called the electron transport chain, and this specifically happens on those folds of the mitochondria. Okay, so remember that those are made of membrane, and this is membrane right here. So this is all about just taking NADH and FADH2 and converting them into ATP. Okay, so if you look up here, NADH and FADH2 go through some reaction. I don't know what it is. They form hydrogen ions. As a release, they get left with NAD. The hydrogen ion there and the hydrogen ions here. Pump through this membrane, build up on this side. So you're going to get a whole buildup of H plus over here. These are protons. So it's called proton pump. Once they build up over here, they're going to go over to this enzyme, ATP synthase, which is going to make ATP. Okay, so they're going to pump through it. As they pump through it, they allow ADP to convert into ATP. So that's the whole point of this, is just getting hydrogen ions off of these to make this. This makes like 32 of the 36 ATP produced as part of that process. So this is a big deal. Again, this is why the mitochondria has all these membranes to do lots and lots of this. Okay? Okay, so that's everything about respiration. For sure, those 3 pictures will be on the test. Okay, so again, you don't have to memorize those. Just be able to look at those and read them, essentially. Okay? Pay attention to what I gave you, like the big picture. What's it do? What's it start with, and what's it end with? Okay? All right, the next thing, that is, not the last, is photosynthesis. So remember that photosynthesis is, we first started talking about plants. Remember that plants have 3 parts. They have roots, they have stems, and they have leaves. So remember that roots are used to bring in water and to bring in minerals and for storage. And they anchor the plant, things like that. Okay, so we looked in lab at roots, and remember we saw a root look like this, and the dead giveaway for this one is these little root hairs that stick out of it. Okay, so those things increase surface area, and they help it bring in the water that it needs. So that's going to be a dead giveaway if you see that cross section. Okay, stems look sort of similar. They look more like this, and the stems, remember the stems hold the leaf up. hold the plant up. They move water up and sugar down. And they do a lot of storage of water and sugar as well. Okay, and lastly the leaves, and I won't have to draw a leaf because there's one right here, but leaves are used basically to make carbohydrates, to make sugar. Okay, so they're going to make the sugar, they're going to move the sugar down, they're going to store sugar and store water, and they're going to bring in gases. Okay, don't let me trick you. Leaves don't bring water in, they bring sugar in and air in. Okay, water comes in from the roots. Okay, so let's talk about the main one here that we need to talk about, which is the leaf itself. So the leaf, remember, has an epidermis. On top of that is a cuticle, which is just a waxy layer on top. Okay, this top layer here is a palisade layer, and that's really important. Probably ask you about that because that's the one where all the chloroplasts are located. So it's got most of the chloroplasts in this top palisade layer. Remember, palisates have columns. So this is where the chloroplasts are located. This next layer is called the spongy layer, and that's really significant because this open area here is all for storage. Okay, so it's going to store water, it's going to store gas like CO2 and H2O. It's going to store sugar even until it's used. Okay, once that sugar and water is needed or used, it's going to go into this vein here. This is a xylem and phloem. Here's the vascular bundle. And that just bundles the vein, which is xylem and phloem, but again, I'll keep them together as xylem and phloem or vein. So that's the vein, is the inside part of this, and that's what's going to move water and sugar. I don't know which one's which. Okay. All right, last thing is down here at the bottom, this hole is called a stoma, and these are important because the stoma open and close to let the plant do photosynthesis. When they're open, it's allowing gas to come in, CO2, it's allowing gas to come out, oxygen, so we can breathe it. So that's a good thing, but the bad thing is that when it's open, we're also going to lose water. So remember that plants have a lot of tricks to do this. Some of them will close their stoma at night. Some of them will just be more efficient and only open it when they have to. Okay? So it's a big deal about losing water. Okay, as a result of this, we talked about pigments. Remember that pigments are molecules that absorb light energy. So as in doing so, we talked about the electromagnetic spectrum and light. Remember that light travels in waves. Okay, light travels in waves, and we looked at the wavelength of light. Here is a graph. This exact chart is on the test. So you can just see that basically from this way to this way, we're looking at increasing wavelength. So this is a short wavelength, is violet, and red is a long wavelength. So VIBGYOR is the colors of the visible light spectrum in increasing wavelength, VIBGYOR. Okay, so we know that in looking at this, then that means that UV light has a shorter wavelength than does infrared light, and so on. And so like these would be the biggest over here. Electric power must be like wow. Okay, cosmic rays must be like that, tiny. Okay, so increasing wavelength from left to right on this chart. Okay, so we looked at pigments, and what we noticed is that pigments absorb light energy. So we know that plants and things are colors because of what they absorb and they reflect. So remember something, a color is what color it is because it reflects that color. Okay, so for example, a blue ball reflects blue light, and a red ball reflects red light, and it absorbs the rest. Okay, so we looked at absorption percent of plants and what pigment they have. So assuming this is 400 and this is 700, we can fill this in. Here's 500, here's 600, So this is VibGyor. Like this. And remember, high is absorb, low is reflect. High is absorb, low is reflect. So this pigment is absorbing, some violets and indigos, maybe a little bit. Okay, it's reflecting blue, it's absorbing green, it's reflecting yellow and orange, and it doesn't even have any red, so we'll leave that off. Okay, so basically you could say that this plant is reflecting blue, yellow, and orange, and it's absorbing Vi and G. Okay, and since that red's not on here, I'll leave it off, but if you were to say it's reflecting red, I could live with it. You wouldn't probably get marked down for that. So, okay, not trying to trick you there. So this would probably be like a blue, yellow, orange. What's blue, yellow? Kind of a bright greenish red plant. Okay? So that's about pigments. So remember that stage 2 is where the pigments actually are used to do something. So this is stage 2 called the light reactions. Okay, and they basically take light energy and convert it into cell energy. Okay, this was the whole oil and gasoline discussion. Okay, so light has got to hit chlorophyll, which would be, two of my my reactants, light and chlorophyll. It's going to get excited, go through that electron transport chain that we spoke of earlier, and it's going to get converted into the first piece of cell energy, which is ATP. So here's a product. Okay, light is going to hit a different pigment. It's going to get excited, go through that electron transported chain, and it's going to form NADPH. So our two forms of cell energy are ATP and NADPH. So those are my products, and my reactants are really light, chlorophyll, ADP, and NADP, if you're keeping score. Okay. Lastly, we know that water is also part of this process. Water gets broken down. So water is a reactant and oxygen is a product. So this is where we get our oxygen that we breathe, and the electrons to run this are also produced here and get used as part of this process. So pretty important that water is given to plants. Okay. All right, last one is stage 3. And so this is called the Calvin cycle, or sometimes called the dark reactions. That just means they don't need light. Okay, so they're sometimes called the dark reactions. So the Calvin cycle is all about making sugar. So this is basically converting CO2 into glucose. Okay, so it's going to take CO2, it's going to convert it into glucose down here. Okay, so it's the opposite of that Krebs cycle where it spun around and broke it down. Now it's going to spin around and put it together. So we're going to input a carbon. We're going to stick it together with some pre-existing molecules, and we're going to knock off that carbon as a carb or a carbohydrate, and eventually, after enough turns, we'll make glucose. Okay, so this has got to spin around 6 times to make glucose. Okay, each time we spin around, we're going to make one carb. Okay, remember glucose is 6 carbons. Okay, we're going to use lots of energy and lots of energy to run this thing, so that ATP and that NADPH that we just made are going to be used to power this. So that's why the cell had to convert light into cell energy so that it could run this wheel. As far as reactants, here's a reactant, here's a reactant, and here's a reactant, and here's a reactant. So if you had to look at all the reactants are there. Product-wise, yeah, I know ADP and ADP are made. Not a huge deal because they're probably going to get reused again. Big deal, this is our main product that we want to focus on. Okay? This is happening inside the chloroplast, inside the stroma of the chloroplast. So here's that chloroplast. So here are the parts. Remember, the whole thing's the chloroplast. This is called a thylakoid, or in this case, it might be pointing to the membrane of the thylakoid. A whole stack of thylakoids is called a granum, but this could be individually too. It could be one thylakoid. And then the liquid outside is called the stroma. So this is the liquid outside here. Okay. So photosynthesis, the opposite of respiration, stages 1 and 2. So pigments and conversion of light energy happen here, and then stage 3 is going to happen here, whereas the respiration started outside and moved in. It went outside and then to in. These go inside out. So again, just keep in mind that photosynthesis and respiration are complete opposite processes of each other. Okay, the last bit of information is about ecology. So the first thing that we did with ecology is we started looking at biomes. So remember, a biome is a region with a specific climate and specific ecosystems. And I'll cover those words in a second. Okay, but it's a specific area on the globe that has a certain climate and has certain ecosystems that all work together. Okay, so we talked about many major biomes on Earth, and these are here. This chart is not on the test, but the information about it is. So it was a good review for me just to say, okay, think about the polar ice caps. They're cold, and they're found in the north. The north and south poles of the globe. Okay, the tundra is found just below that. Has similar conditions, very cold. Has permafrost. Kind of a word to help remember that, permafrost. And very few plant species. The polar ice caps only had lichens growing there, and those are just algae that grows on stuff. Permafrost, you're starting to see some small bushes and things like that. Okay, the boreal forest is below that, so now we're starting to get into a little bit more of a temperate region, even though the boreal forest is really cold. Okay, you're going to see lots of conifers here, lots of pine trees, things like that, and cold weather animals. Okay, this is northern Canada. As we start getting warmer, we also then start worrying about dryness and wetness, because that is going to affect what biome it is. So we didn't really talk about cold and warm deserts, so we talked about the prairie, and we talked about the deciduous forest. So Lower Michigan is a deciduous forest. That's where we have trees like maple trees that lose their leaves every day. And so we're temperate. We have 4 seasons. That's what temperate basically means. Okay, prairies, we talked about the grasslands of Kansas for this one, and so basically it's a temperate region too, but a little bit drier than what we get. Okay, again, getting warmer. The warmest, driest place is your deserts, and so we talked about the Mojave Desert and the the Sahara Desert. And then we start getting warm and wet, and so now we start getting the tropics. So remember, the tropics basically is along the equator. Okay, and they have 1 or 2 seasons at most, and it's basically just rain. The savanna didn't have as much water, and they're located in the outer skirts of the tropics, and that's warm and dry, kind of like the scrub lands of California. But again, as we get wet, we get tropical. Okay, so that's a pretty good review of the biomes and where they're at. So this globe is pretty good too, because you can use this to practice. So here's my polar, here's my tundra, here's my boreal forest, here's my coniferous and deciduous forest, here's my grassland, here's my scrub forest over here. Now we're starting to get into the tropical zone here and here. So here's the equator. So this is my tropical rainforest zones here. And then we start getting back into this southern temperate zone. And so we start seeing a repeat of some of the stuff, deserts and things like that. Here's Antarctica down here. So that's South Pole. Okay, some of these lines. Here is the equator. Tropic of Cancer. Remember, Cancer is on top. That's 23.5 degrees north latitude. Capricorn is the same thing, but it's 23.5 south latitude. Okay, so that's here. So this is the tropical zone here. C and D are tropical zone. B and E are temperate. And so now we get those 4 seasons. This is the Arctic Circle, and this is the Antarctic Circle. And those are at 66.5 degrees north and south latitude, respectively. Okay? Okay. So we had some terms that we looked at. We looked at the difference between like populations and communities. Remember that a species is where 2 organisms are similar and they can breed and make offspring that are the same and that are viable, can reproduce and have more. A population, see how good of a stick drawer I am. A population is where you have a whole population of the same species. So we got lots of little stick figure guys, and they're all basically the same, and they're sharing an area. Okay, so that's a population. A community is when you have several different populations. So you have people, and you have deer. Oh, Dustin Roberts will be mad about my picture of a deer here. So you have deer, and you have chickens, and you have trees, and you have, everything else living in an area. So that's a community. Okay, several populations of living things. Okay, then we have an ecosystem. Ecosystem is all the living things and the non-living things. So remember, living is biotic, non-living is abiotic. Okay, so that's an ecosystem when you add living and non-living things together. And then lastly, we talked about a biome already, so we know what that is. That's just several ecosystems plus climate. Remember that climate is annual weather, and weather is day-to-day. So, climate really determines your biome, not your weather. Your weather is a day-to-day thing. Your climate is an average number, okay? And then lastly was the biosphere, and the biosphere is basically the Earth, everything on it and above it, okay? I do know that I put a couple of these terms on the test, just asking you what is a community, or, give you a definition, pick the word, okay? So a couple of these are on here, all right? Last but not least, I talked about competition. So we have predation. And predation is when basically one thing eats another thing or preys on another thing. So that's a type of competition where one wins and one loses. Okay? Predators and prey. There was also a thing called symbiosis. Okay, symbiosis is when things share a niche. A niche is basically your role in life or your spot. So symbiosis, there's a couple different kinds of symbiosis. One was mutualism, and that's when both are affected, both are helped. So it helps them both. So like barnacles attached to a whale, and they keep the whale clean, and it gives the barnacles a ride. So that's a good thing. We also have commensalism, where one is helped and the other one is not helped or hurt, just they're part of it, and whether they want to or not. And then, of course, the third one's predation, where one wins and one loses. Okay? So, I kind of remember this one, and then I can work my way around to this one. Okay, all right, last topic was about succession. And succession is just a fancy word that basically says that like ecosystems will recover. If you cut down all the trees, they'll eventually grow back again. So we looked at 3 different kinds of ecosystems: an old field, and that just means like take a farmer's field or a football field and stop mowing it for 100 years. What'll happen? We have a thing called a pioneer species. And a pioneer species is going to be the first thing that inhabits an area. Okay, now this could be after a disturbance like a fire or something like that, or it could be new ground. So we'll just assume that this is secondary succession. Secondary succession happens after a disturbance. So I'll say it's either a forest fire or a lava flow, something like that. So the pioneer species for an old field is going to be probably a grass, something like that, maybe a lichen even. Okay, it's going to end up to forming a climax community. That climax community for an old field is going to be a forest, probably a deciduous forest, but it depends on what biome you're in. Okay. So you always start with a pioneer species, you end with a climax community. In an old field, like our football field, it'll start with grass, weeds, and it'll end up as a forest. Okay? A pond. If you take a pond and clear it out, pioneer species is probably going to be grass, or it's going to be maybe some kind of water plant, like a water lily, something like that. And eventually, believe it or not, the climax community for a pond is also a forest. If you watch all ponds, eventually they turn into a forest. All the water's gone and it's all trees. Okay, last one was beach succession, like if you go to Lake Michigan. The dunes, eventually you see the dunes form as a pile of dirt. Then you're going to see like a little bit of grass growing. And as you walk further and further away from the lake, you're eventually going to see guess what? Forest. So all 3 of these things are going to turn into a forest as their climax community. Okay? Primary succession is a little bit different in that primary succession is basically new ground. So that either means like, lava flow, hard ground, or something like that. Usually that pioneer species is going to be a lichen. Okay, so, all right, that's it. Hope you do well. Peace.